Hi everyone. We're going to start talking about the lab that we're going to be doing. And here is the pre-lab. Here's EMG. Here shows the actual lab. So you guys watched the other two videos that was talking about muscles and muscle contraction. So muscles, skeletal muscles, do not, I repeat, do not contract on their own. They need to be stimulated. What's doing the stimulating? Nerves. Yes, nerves, neurons. So neurons are nerve cells. So if I told you right now, clench your fist, where is that control coming from? Your brain heard, well, your ears heard the sound, took the information in your brain processed, okay, clench your fist. Do I want to do it? Do I not? Okay, let's say you do. So then action potentials, or those are the nerve impulses, or those are truly electrical impulses, will send information down your arm to your muscles, and then we clench our fist. So a lot of electrical signals are happening. It's happening so fast. It's milliseconds. It's thousands of a second. So that stuff is going on in order for you to contract and clench that fist. So today's lab, Biopack, okay, this is what we're going to use to actually conduct or pick up those electrical impulses, okay? So this says Biopack. This shows an arm. We're going to put electrodes on the arm, hook it up to a computer, and then these are actually millivolts. So we'll see the changes, the actual electrical changes that are coming off of the skin, okay? So here's an introduction. Oh my gosh, we've heard this now how many times? Types of muscle, so cardiac, smooth, skeletal, right? So cardiac in the heart, smooth around hollow organs, skeletal. Today's lab, we're focusing on skeletal. So here, we've seen this too. Human skeletal muscle consists of hundreds of cylindrical shaped cells called fibers. The reason, I had so many questions about this is it can get so confusing. A cell is the same as a muscle fiber. A muscle cell is the same as a muscle fiber. So they're bound together, da 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 da, and then we have the muscle, okay? But in reality, it's neurons that will come over and then they will excite the muscle and then the muscle will contract. So it says axons are long cylindrical extensions of the neurons. Axons leave the spinal cord and the brain, okay, da da da, and they go to the appropriate skeletal muscles, so this shows that. Although a single motor neuron can innervate several muscle fibers, each muscle fiber is innervated by only one motor neuron. So that neuromuscular junction, there's the neuron and then there's the muscle. And then the neuron sends the information and that will make the muscle contract. So we were watching those videos. So, all right. So now in lab, you're gonna clench your fist, then you're gonna actually clench it harder and harder. And it's like, okay, what's happening? With muscles, we can have an action and we can actually increase the strength, okay, of our contraction. So that is called motor unit recruitment. It says when an increase in the strength of muscle contraction is necessary to perform a task, the brain increases the number of simultaneous active motor units within the muscle. This is known as motor unit recruitment. So this is something that we are going to measure in today's lab. When muscle's just sitting there, it actually is doing some motion too. It's doing some contraction, I should say. It's not moving, but it's contracting. Resting muscle in vivo, meaning in the body, exhibit a phenomenon known as tonus, a constant state of slight tension that serves to maintain the muscle in a state of readiness. So we're going to pick that up too. We're going to pick up tonus. And then the biggie in the lab is electromyography. EMG, is electromyogram. and electromyography is the detection, amplification, and recording of changes in the skin voltage. So we're really picking up the electrical changes that are happening off of your skin, produced by underlying skeletal muscle. The recording is the EMG. So what are we going to do? You do not, I repeat, you do not have to print out all of these pages, okay? This is just for you to use to look at what we're going to do. And I would look at the fast track. When you guys come into the lab, I'll show you how to plug in the electrodes, okay? They're electrode lead sets. Here it shows you, I'm gonna zoom in a little bit, okay? Here it shows you where we're gonna put the electrodes. So I'll show you in the lab also. We're gonna put 2 electrodes here, 1 here, and then the electrode lead set, it actually shows you where you're going to connect them. The red, the black, and the white, okay? And so at first, we're going to connect, let me see, what does it say? To the dominant arm. So if the person's right-handed, that would be, the right would be their dominant, okay? And so we're gonna look at that. And then we'll start the Biopack, okay? First, we gotta do calibration. I'll show you guys how to do that. And then on the computer screen, you know, we'll see a little line. That little line is actually showing the electrical changes. And then you clench, it says to click calibrate. We got to calibrate the machine. You clench your fist as hard as possible and then wait for the calibration to, then release, then wait for the calibration to stop, okay? Then we're going to do something, it says detailed explanation of recording, segment 1 records dominant, segment 2 records non-dominant. So we'll do dominant and non-dominant. So it says perform a series of 4 clenches, hold for 2 seconds, release, begin with a weak clench, and then increase equal increments so the 4th clench is at the maximum. So again, the whole point is to do recruitment and to pick up the electrical changes that are happening. So you'll click, you know, start, you do a little clench, relax, a little stronger clench, relax, a little stronger, relax, and then the maximum. It says clench, release, wait, repeat, okay? You can review your data. If it looks good, you can keep going. If it doesn't look good, you can redo, okay? You can redo. Then we'll put the leads on the non-dominant arm and we will do the same thing. You click resume. We do exactly the same thing. Perform a series of 4 clench hold, click suspend, and then click stop, okay? Now that is all, and we'll see if we have more time. If we have more time for the lab, we'll do it, but you know, with the setup, etc., it might be all. If not, we could do something called fatigue, which is really cool. Like fatigue is a real thing, you know? Imagine if you are lifting a couch and you're holding it and holding it and holding it, and after a while you're like, I'm too tired, I'm fatiguing. That is a real phenomenon. We can actually do it with clenching, and we'll see. If we have time, we'll do it in the lab. So then we are going to analyze the data. This shows how we do data analysis, and I'll actually show you. You would click, see that little bar? You would click on that little bar, and then you can highlight a section in the graph, and then over here, it'll show you the changes, and it's going to be in millivolts because we're actually looking at electrical changes. And so we can record that. I'm going to go to this page. Now, these are the 2 pages you need to print out. I would print out page 11 and I would print out page 12. Now, if I go back, okay, so we're going to do the student profile. If we have time, we'll do it on multiple students. Let's see what it looks like. We could do the clusters, the dominant arm, what's the change with the mean with the millivolts, and we'll take a look at that. Then the non-dominant arm, you will get the mean for all of them. percentage increases. We'll talk about that. Tonus measurement. Tonus is like when the arm is just sitting there. And then we have some questions about the lab. Okay. So I wanted you guys to look at this. Whoops. We'll go down here. I wanted you guys to look at this, and I wanted you guys to have this video first. So when you walk into the lab, you know exactly what we're going to do. And then I will have, you know what? I got to do this. I will have the Biopack set up. You'll be ready to go. That way we can do a few instructions on what to do, and then you guys will get right to it. So pretty exciting. We're going to look at muscle. We're going to look at action. We're going to measure the electrical changes that are caused by action potentials. Okay, so for the lab, again, only print out the last 2 pages. See you in lab.